morning, Your Excellencies, ladies and gentlemen, and welcome to the second day of Wish 2022. It's been great to see the discussions happening on social media and live here at Multaka, and I hope you're feeling energized about the second day of the summit. Just a quick reminder that we will continue to encourage everyone here and also those who are watching remotely to continue to engage both through the app and via social media. And don't forget to use the hashtag WishQatar2022. Now, first up on the main stage today, it's my pleasure to introduce Giles Duley, who's a photographer, writer, presenter, and CEO of Legacy of War Foundation. Partnering with leading global humanitarian organizations and charities, he travels the world to document those facing challenging and at times horrific humanitarian situations. Yet he also manages to capture the strength of those who fight adversity rather than succumb, ultimately providing a message of hope. We're honored to have some of his work hanging in the Innovation Gallery downstairs, and I'd really encourage all of you to go down and have a look today. Please, can you join me in welcoming Giles to the stage?
Good morning, everyone. I must admit, I was in the conference yesterday, and as a humble photographer, it's somewhat intimidating knowing you're going to be speaking to some of the most brilliant minds in the world. And I must admit, at 2 o'clock this morning, I still wasn't actually completely sure what I was going to talk about. I'm a storyteller. You know, I've always said I don't think a photograph can change the world, but I believe it can inspire the person that can. And that is my role, is to tell stories. As I say, I'm not a particularly smart man. I didn't study at university, but I've been very lucky in my life in that I have been given two gifts. One is all the stories of the people that I meet and the knowledge I have gained from them. And secondly is my own life experience has enabled me to see the world in a slightly different way. So today what I want to do is kind of talk about some of these stories. And really for me, it's almost like a jigsaw puzzle in life. And it's only when I put the pieces together and I step back, like I did at 2 o'clock this morning, that everything makes sense. I wanted to talk about the photographs that are here exhibited, that Wish very kindly supported me to do some stories that we could share here. And I'm going to start with those stories. And I'm also going to talk about this word that gets used a lot, resilience. I have my own take on what I believe resilience is. I believe it's a superpower that we don't take full advantage of. So, stick with me. We'll put the jigsaw together, and hopefully by the end, it'll make sense to all of us. So I'm going to start by telling some of the stories that you can see in the exhibition downstairs. The first set of photographs you will see down there are taken this year between February and August in Ukraine. I have been visiting Ukraine for many years, documenting the wars in Donbass, and obviously in February of this year, with the Russian invasion, I returned to document the impact of the war on the civilian population there. I traveled across the country, and as I say, you can go downstairs and see these photographs and read some of the captions. I wanted to focus on a couple of the stories of people I met. This is Yasha. Yasha was at home in his house just outside Odessa. It's a relatively small village. The family had decided to remain. One member of the family, a cousin, she decided to leave, but the rest decided, because the area was no military value, that they would be safe there. Unfortunately, one day, a rocket hit the apartment building where they were living. Yasha was very badly injured. He has multiple fractures across his legs, his arms, but his parents were killed in that attack, and actually he was trapped in the rubble over his mother's body. for 6 hours until they could get him out. But it's not just Yasha that was a victim of this attack. And often there are people affected by conflict that never appear in statistics. I said one of the family had left. It was a cousin who had been brought up in this house. She had gone to Poland. She was 24 years old. She was living there as a refugee, and she was watching the news one day, and she realized they were talking about her village. As she watched the news, she realized they were talking about her apartment building. As she continued to watch, she recognized the front door. She realized that it was her family that had been killed in this attack. She returned because the only two survivors were Yasha and his brother, and she now has taken full guardianship of these two injured children. A young girl, really, of only 24, now having to take over the lives of these two children who are both living with severe injuries. These are the impacts of war we don't see in statistics. But war affects everybody in those communities. Yasha, I have to say, this is a particularly moody photograph of him. He'd just been doing his physiotherapy. He does have his lighter moments. But for him, he is living in that moment of trauma. Most of the time, when you talk to him, you realize he's not even looking at you. At the moment, he is living really in his own mind. It's hard to imagine what a boy of 8 years old is processing in his mind at the moment. Now, when the war actually started in Ukraine in February, I was actually in Gaza. I was doing a story in Gaza on men who have had amputations caused by traumatic injury. Most of the injuries are caused by bullet wounds, where their legs were targeted on purpose, leaving them with severe fractures, meaning they had no choice but amputation. Many of the men choose not to have amputations. They go for years in chronic pain because there's obviously a lot of stigma associated with amputation. So I was doing a story, and again, you can read further all these individual stories downstairs about various people that had gone through this experience. But I wanted to talk to you about my friend Amro. Amro had an injury caused by a bullet in his leg. He had gone to the Great March with a bunch of his school friends. He was 17 years old. They were taken by bus. When he arrived there, within minutes of getting off the bus, he had been shot in the leg. When he woke up, he found that his leg had been amputated. He was taken to Turkey for further treatment and then returned to Gaza. When I met him, he had not left his home in 4 years. Why? Because he said he felt ashamed. He was so embarrassed. He said, everyone will look at me, people will judge me, people will look down on me. I don't want to be seen in public. You know, imagine this boy who was 17 at the time, who's now 21, for 4 years, too afraid to leave his flat because of stigma. Now, I sat with him. I will be telling a little bit about my own story, but I'm a triple amputee myself. I said, Amro, if we go out and have a coffee, you're missing one leg. Trust me, they'll be looking at me. They'll be looking at the white guy with 3 limbs missing. He laughed and he agreed, and we decided we would go out and we would have a coffee. And we sat down, and I tried to sort of repurpose him and try and think things differently. I said, maybe when they're looking at you, they're admiring you. Maybe when they look at you, they see something in you that they don't possess, a strength, a resilience that nobody else has. I said, reframe it. When they look at you, be proud. When they look at you, sit up straight and acknowledge that they are looking at you, and nod your head and say thank you. So we talked about it a lot, and I'm glad to say that since then he has been going out regularly. He's joined Amputee Football Club, and his life is progressing. And then I'm going to move on to a final story that's, again, downstairs. It's kind of a little preview for the exhibition. You can go down and see these 4 stories. It's a set of photographs that I did actually a few years ago. A set of photographs that means a great deal to me. It's a group of women from DR Congo who are living as refugees in Angola. Now, I'm not going to tell the specific stories of these women, but they were some of the most horrific stories I've ever heard, of sexual violence, of attacks against their families, of persecution of the villages, destruction. But when I met them, I did not see the suffering, I did not see this. In fact, I met one of the most amazing group of women I've ever come across. I'd gone there with a UN organization. and I say this completely off the record in front of everyone, but normally when I go with an organization, there's a list of people they want me to meet. I always sneak off. I always kind of sneak out the back and find people myself. And normally I wander around a refugee camp, as I did on this particular time, and as I wandered around, I found a group of women. I went over and I said, "You look like trouble." And they laughed and said, "Yes, we are the ladies that are trouble in this community." Auntie Rose was the matriarch, incredibly strong woman. We sat, we laughed, we chatted all day. I have a rule. One thing that was missed at the beginning of the introduction is I'm also a chef. And I have a simple rule that I will never photograph someone if I haven't eaten with them first. For me, it's a sign of respect. It's a sign of building a relationship. So we sat, we shared this meal. We were laughing, having a lot of fun. In fact, it was at one point, Rose's children arrived, and they looked at me a bit confused. I'm not sure if they'd seen a white man before. And they said, "Who is this?" And she said, "Oh, it's my brother." And you could see them kind of doing this double take going, not quite figuring it out. But it was a day of celebration of life. And I asked the women, as I always like to when I'm working, how would you like me to photograph you? How do you want to be represented? And I was showing them a few photographs that I've done in the past. In the long past, I used to be a fashion photographer. And I was showing some of them these photographs, and they said, "That's how we want to be photographed, in the same way that you photographed these people when you were working for Vogue and all these magazines." I said, "Fine." I went back the next day. We had an agreement that we were going to have a party to celebrate, to do this photo shoot. They got rid of all the men. All the men had to leave. The only thing I had to bring was some batteries for the radio, so we had music. And we had this amazing day doing these portraits. We had this white sheet that we set up, and I did these portraits. And it was a real celebration of life. And I came back, and those photographs were published on the front of a fashion magazine in Los Angeles. And we were able to send a copy of this magazine back to the women. Now, why am I particularly talking about these women? These women taught me what resilience really is. I remember sitting with them. We were celebrating life. We'd done the interviews about the stories, but we were having this day, doing the photo shoot. We were really, as I say, enjoying the day. And I remember asking them, how was it possible? How could you still celebrate life after all you've been through? And they looked at me and they said, "Giles, we are here because we are strong." And then they said, "Do you know why though we are celebrating today?" And I said, "No." And they said, "Because of you being here. Because in you, we see that you have suffered. But we see that you have come through the other side, and we see your resilience." And so I decided that with these portraits, I wanted to celebrate them in a different way. So I have a friend who's an artist, and we created these pieces of work that celebrated the women beyond just the portraits, because there is something about them as if new life had come from them. So we celebrated with flowers, with a celebration of life. This is, the picture's not coming up. Getting no photographs? Oh, here we go. So here are the photographs of the women that we created, these beautiful artworks. This is Jermaine. Jermaine had one of the most incredible stories that I think I've ever heard. She had been escaping from DR Congo with her family. She has 6 children. Her oldest was only, I think, 8, 9 years old, and she had a baby. The 8, 9-year-old was carrying the baby as they were crossing a bridge. There was this huge amount of people trying to cross the bridge. There was fighting very nearby, so people were in a panic. When they got across the bridge, she was looking for all her children. She saw the 8-year-old, but the 8-year-old did not have the baby anymore in her hands. The child was crying, and she said in the push, the baby had been dropped. Now, I asked Jermaine, obviously, how did she feel about it? And she said, how could I be angry with her because she is just a child herself? But of course, for the child, for the family, this was devastating. They managed to make it to the refugee camp, but the baby had long gone. 6 months later, though, she was sat at a tent, and a friend came by saying, I just walked past the orphanage, and I saw your baby. And so they went, and they found the baby, and the baby was reunited with the family. But again, as I say, Jermaine was this incredibly strong woman, only in her mid-20s herself, full of this life. Now, as I say, we put this exhibition together with these 3 stories, and it was only last night that it all made sense to me. Often I do things through instinct and don't really think about it. I'm going to come back to it and explain it in a minute, But now I'm going to tell you a bit about my own story and how this all links together. I told you it was like a jigsaw. Trust me, the pieces will all come together at the end. Now, in 2011, I was working in Afghanistan. I was there documenting the impact of war when I stepped on an IED, an improvised explosive device. I lost both my legs and my arm. I remember waking up. I was lying on the ground. I thought these would be the last moments of my life. I was picked up by an American medevac crew and flown back to Kandahar, where they did emergency surgery, and then flown back to the UK. But actually, I don't really want to focus on that part of the story. I want to talk about what I call ultimate lockdown. Now, I want you to imagine this for a minute. When I woke 3 days after my accident, I was in an intensive care unit in the United Kingdom. A room that has no windows, where the lights are on, people rushing around, the sounds of machines. I didn't even know which country I was in. If you imagine the panic of waking up like that, I did the obvious thing you want to do. I wanted to scream. But I realized I couldn't because I had tubes in my throat, in my mouth, even cut in here. I tried to move my one remaining hand, but it was in a plaster cast because it was broken and strapped to the bed. I realized even my head was strapped. I soon realized that the only bodily function I had control over was blinking. In fact, it was 3 days before anyone even realized I'd regained my consciousness. For the next 46 days, that was the only way I could communicate with the world, was by blinking. Now, at first, I was in a panic. It was like being thrown in the freezing cold water. I couldn't think straight. I was going absolutely crazy in my mind. But I knew that if I was going to survive this, I would have to find a way to create some kind of world in my head, some kind of discipline. So I created projects. I created various exercises. I imagined past events, things I wanted to do in the future. My favorite project was called 100 Portraits Before I Die. I imagined the 100 people I wished I'd done photographs of in my life, and I'd visualize those photo shoots. I'd imagine turning up, meeting the person, whether we'd shoot in a studio on location, the camera that I'd use, black and white or color. And I would see the photograph at the end. I would visualize it so clearly. And I would critique it and think what I could do differently the next time. And you know what's amazing about the human mind? Is that if I look at how I worked as a photographer before and after, my technique and everything has completely changed. Because the human mind means even when we are so locked down that we can only communicate by blinking, we can improve ourselves as people. Now, 3 months after that injury, I was well enough to be moved into a high dependency unit. And for the first time, I was put in a wheelchair and taken to have a shower. It was the first time I saw myself in a mirror. And I was repulsed. I was disgusted. I was shocked. It was the first time I really came to terms with what had happened. My missing legs, scars across my body, my arm. It was a moment I realized what my future was. I was told I would never walk again, never live independently. I would never work as a photographer again. Everything I valued had been taken away from me. I went to bed and I cried myself to sleep. And I remember thinking I wished I'd just passed away on the helicopter when I was rescued in Afghanistan. I was not brave enough to deal with this new reality. But the next morning, something changed. I'm notoriously stubborn, and the next morning, my stubbornness came back with a vengeance. And when I woke up the next day, I said, from this moment on, I will never think about what I can't do, but I will focus on what I can, and I will excel at that. And that became a mantra for life. And it didn't just help my recovery, but it's now helped me in every aspect of my work, from the charity that I run, to my photographic work, to even working as a chef. Because each day I repeat that to myself. What can I change today? What can I focus on? Don't worry about the things I can't. In fact, if I was to ask most people in this room, what is the thing bothering you the most? I can guarantee that the majority of you will tell me something you have no control over. It's important that we're aware of those things, but if they're at the forefront, they block us from being able to do anything. So for me, whether it be COVID, the war in Ukraine, those things are in my periphery vision, and each day I focus on what I can do and try and excel at that. The first thing I needed to do was to re-examine how I saw myself. So I decided to do a self-portrait. And this was an important moment in my recovery. This was 3-4 months after my injury. I went to a photographic studio and I did this photograph. Because this was the moment that I put that mantra into action. Because this is the moment I said, actually, I do not care, in the nicest possible way, what you think of when you look at me. Because what is important is how I see myself. And I knew when I took this photograph that, yes, I was missing limbs, but inside, I was more focused than I'd ever been. I was stronger than I'd ever been. I was a better man than I'd ever been. What I didn't realize is at that moment, I had discovered my resilience. Now, I said I wanted to tie in these stories. And I realized that when I was putting these photographs together for the exhibition downstairs, in many ways, what I was doing in the 3 different stories was reflecting the 3 stages of my own recovery. In the first photograph of Yasha, he is still in that moment of trauma. The moment for me where I spent 46 days locked in my own mind. The moment when you think there is no hope, there is no future. With Amro, he is going through the stage of acceptance. He is going through that process that I went through when I came to terms with what had happened. And in the picture of Jermaine, we are seeing the flower of resilience blooming. I don't believe we can make ourselves more resilient. When 2 years ago, COVID happened, people rang me up and they said, Giles, come and talk to us. We need to be more resilient, whether it be a business, an individual. And at first, people were surprised when I said, you can't make yourself more resilient. Instead, I believe resilience is life's gift. It is the gift that life gives us for those difficult times, for our suffering. Each one of us has that experience where the elasticity of our experience stretches and we become more resilient through it. Some of us have gone through very extreme moments that have given us even greater superpower of resilience. And that is what I celebrate here today and in the exhibition downstairs, is that gift of resilience. And one final thought. When I set up my own foundation, the Legacy of War Foundation, people said, why are you doing this? There's so many great NGOs, which there are. And I said, because there is one asset. There is one thing that I don't believe that NGOs are using enough of. And what is that asset? It is the resilience of the people they are working with. The people often referred to beneficiaries have this gift of resilience. And what I wanted to do was to nurture that. Because I don't believe we can empower anyone else. Often I hear NGOs saying we empower people around the world. I know when I was in hospital, nobody empowered me. But what people did is they broke down the barriers that were stopping me enabling myself. And that is what we do as an organization, is we work with people's resilience. We celebrate that resilience. We do not empower them, but we break down the barriers that stop them empowering themselves. So if I have one bit of advice to anybody working here, is look at the beneficiaries, look at your patients, look at the people you're working with, and realize they have a superpower that you don't have. And it is our job to make sure that superpower of resilience is allowed to fully flourish. Thank you. Thank you.
Wow, thank you, Giles. Thank you so much for those powerful words, truly remarkable and very, very inspirational. We now would like to shift our attention to scientific progress and innovation. Now, yesterday, my colleague Stephen Saka was able to sit down with the co-founders of BioNTech, Professor Özlem Türeci and Professor Ugur Sahin, to discuss their journey in developing the first mRNA vaccine targeting COVID-19 and where we can expect the next big innovations in healthcare. Let's watch.
Professors Ugur Sahin and Özlem Türeci, it is a real pleasure to welcome you both to the WISH conference. Welcome.
Morning.
Thank you.
Morning,
Thank you
Stephen.
for having us.
Well, we really appreciate your time. And of course, this conference is the first that we've held face-to-face in Qatar at WISH since the COVID pandemic. And I don't mean to imply it's all over, but nonetheless, here we are back together again. And it's a great time to reflect on what happened over the last 2 and more years. So let me begin with a simple question. You two were world-renowned researchers whose main specialism appeared to be cancer research. And yet here you are now seen as two of the great pioneers who developed the first mRNA vaccine available to the world. How did that happen? Ugur and then Özlem, give me your thoughts.
Yes, Stephen, the story, of course, started with the outbreak in Wuhan. And we realized early on that the outbreak in Wuhan will become a global threat, a global pandemic. At that time point, we had a technology in our hands that we had developed over the last 20 years. And the two features of the technology made us believe that we can contribute to develop a vaccine in the shortest possible time. One feature was that we had new technology, mRNA, that allows to make extremely in a fast fashion a vaccine. And the second aspect was that we knew that our technology is able to induce extremely strong immune responses, and that is what you need in a pandemic outbreak situation, a technology that could allow you to come up with a vaccine while the pandemic is ongoing.
And Özlem, let me address with you something just mentioned by Ugur, which is speed. Because obviously speed was of the essence, but the world, when the pandemic first became visible, the world was worried because, frankly, in previous vaccine rollouts, we've seen it take 10 years and more to get from the initial science, the lab work, through trialing, through the complex approvals process, to actually get a vaccine rolled out and produced. We couldn't afford to wait 10 years. How confident were you from the very beginning that you could make this much, much quicker?
The speed, why we were able to do that has several reasons. One reason is that we had a technology which is very versatile, the mRNA technology. And it was clear to us from the very beginning that with this technology, certain steps can be much faster. For example, when it comes to manufacturing or lead candidate selection. And we had exercised that many times in our cancer trials. And we could, so that the entire team and the processes were optimized for speed, for personalized cancer immunotherapy, where you are with each personalized cancer vaccine in the race against the patient's, the individual patient's tumor, you need the speed. So speed is in principle integrated or intrinsic to the technology. This is one. And the other is that we started processes which normally in drug development are conducted sequentially in a parallel way. For example, we prepared for the phase 2, phase 3 trial during conducting the phase 1 trial and ensured that those topics which we could not address because the data was lacking and had to come from the phase 1 trial, that we had placeholders for those. And the third reason why we could be fast was that all stakeholders had the same joint sense for urgency. So, for example, regulators reacted promptly. We had rolling submissions on which they worked even before having the full data sets. And these aspects coming together allowed to be fast, even though we did not cut any corners.
Do you think, to use a terrible cliche, this is the new normal for the rollout of future vaccines, that we can expect that the sort of speed that you operated on, and maybe, if we're being optimistic, to see the speed increase even more? What do you think?
I don't think that this will be the new normal. We have to understand that under the very extraordinary situation created by the pandemic, all those stakeholders who have to act together prioritized the vaccine development ultimately and focused on it. It was on the top of their to-do list. Everyone, including, for example, regulators, worked in non-regular working hours. So that might not be achieved in peace times.
Just a thought about your technology, because of course you were pursuing, as you said, your mRNA approach to the vaccine. There were others who were using much more traditional vaccine approaches, and you sort of all arrived at your own vaccines. Do you think that the world was initially suspicious of the new technologies you were applying, and do you think that suspicion is now completely gone?
So every new technology, every kind of medicine which is newly introduced, of course, comes with the requirement to inform people about how this technology works. We had chosen more than 20 years ago to work with technology because it comes with excellent safety features. So messenger RNA is a molecule invented by nature. It is part in all of our cells. And in our cells, it takes the function that it transports the information from the nucleus of the cells, where the information is wired in DNA, transports the information to the cytoplasm, and this information, the cytoplasm, is used to instruct the cells to produce proteins. And the very nice aspect of mRNA is that after it fulfills its function, it is completely degraded. So it disappears from the cells. And this gave us the confidence that having a technology that we can use to instruct our immune system by bringing this mRNA from outside into the cells, and thereby instructing the cells to make their own vaccine. And after it has produced the vaccine, it disappears, would be an extremely safe approach. And of course, this requires, we worked on this technology more than 20 years, addressed all kinds of potential risk. That's what you do as a scientist, technology developer, and drug developer. We had all this information, and we started to communicate this information. And happily, also our colleagues and the scientific community started to communicate the features of mRNA. We saw more and more, for example, YouTube videos explaining the key aspects of the mRNA technology. And the surprising aspect was that, of course, there was at the beginning a hesitancy. So people preferred at the beginning, for example, in the time of September, October, people preferred when a vaccine is available to take a classical vaccine. But this changed dramatically after 3 months. And most people now prefer to take an mRNA vaccine because they are informed and because this technology is now applied to more than 1 billion people worldwide.
Right. Now that's fascinating what you just said, that you feel now people have embraced the mRNA technology to the point where they actively want it more than the more traditional vaccines. But of course, for you two, having created this new vaccine, having proved that it worked, you then had to watch the world try to roll it out in the most effective way possible. Because for it to be truly effective, it's not just a question of the science and of developing the vaccine, it's then making sure that it gets into people's arms. Now that we can look back a little bit, what do you make of the way the global community has handled the rollout of the vaccines? And I'm particularly thinking of the way it has or has not achieved some degree of equity in the degree to which people have availability of vaccine. Give me your thoughts.
So at the beginning, what we realized when we got the efficacy data for our vaccine, we had planned to produce about 1.3 billion doses in 2021. That was the original plan, assuming that many other companies would also come up with vaccines and that we together could supply sufficient doses worldwide. And we realized then that other vaccine developments were delayed, yeah, and we realized that much more vaccine doses are required. So we revisited our plans and asked the question together with our partner Pfizer, how to increase the production scale that was originally planned, and this originally planned production plan was already really challenging. But the teams came up with ideas how to increase the amount of mRNA per batch, how to produce more batches, how to acquire additional sites. For example, we acquired a manufacturing site in Marburg in Germany, and were able to set up the new technology within 6 months there. And we increased the production scale of more than 3.4 billion doses that were then delivered in 2021. But still, we realized that the vaccine, particularly in the first 6 months, of course, was not available in a way that everyone could get access to the vaccine, because the demand for vaccine doses in the Western world, in the United States, in Europe, was much higher, yeah, than we could deliver.
I mean, to put it bluntly, the governments in the world that had the resources and the money were ordering vast amounts of vaccine to make sure their populations were vaccinated not just once, but twice, and sometimes with reserve stores as well. And while that was happening, the poorer world, the emerging economies, the developing world, particularly if one looks at the continent of Africa, many of the countries in Africa were really not getting access to the vaccine, and their populations were largely going unvaccinated. I wonder, Özlem, whether you looked at that and felt deeply anxious about the way it was working.
Yes, Stephen, we were newcomers to this field and were not aware of the mechanisms which would come into play in the case of a pandemic and how the world and the systems we have set up would work. And when we realized that the inequality of vaccine distribution and availability, we felt the obligation that we also had to contribute to address that. Much in the same way how we felt the obligation to go into this field of COVID-19 vaccine development in the first place. And the obvious and immediate thing to do was to provide vaccine at cost to low and middle income countries. About 40% of what we delivered in 2021, and Ugo, correct me if I'm wrong, went to low and mid-income countries, but that can only be the first step. Part of the plan was also to enable people in those regions where a vaccine was not available in an equality manner, to enable them to produce in their regions vaccine for themselves, and that means technology transfer. Now, technology transfer is not an act; it's an attitude. It means to commit, and We have committed to help to build vaccine manufacturing plants based on our technology, which is very lean, and ecosystems, because that also is required in such regions, starting with the African continent, and those plans are very concrete and already in action.
So let me ask you, I mean, because people here at WISH will be very interested in this. What investments are you making right now, and where to address this, issue of making sure the poorer countries have their own production and manufacturing ability when it comes to vaccine rollouts?
Stephen, the challenge that we realized is that particularly in the beginning of the pandemic supply, there is a much higher demand than even centralized manufacturing could enable. And we believe the only way to solve this problem is to enable that different regions on the planet can produce their own vaccine, so that everyone can ramp up quickly and ensure that the vaccine is regionally produced and supplied. So the challenge for that is technology, because as Özlem said, technology transfer requires time. And to build, for example, a new mRNA manufacturing facility somewhere in Africa, and that was our goal, to make vaccines really available and this technology available in Africa, requires several years of construction and implementation and process transfer. So mRNA vaccine production has about 50,000 steps, because it's not just the manufacturing itself, but also every aspect of quality control of all the ingredients, and there's a control system that is extremely complex.
So have you started, Ugur? Have you started, and if so, where?
Yeah. So we asked the question how we can overcome this challenge, and for that we developed a new way of manufacturing, which we call biocontainer concept. The biocontainer concept is based on the idea, combines two general ideas which are coming from the IT field. This is the copy-paste approach, and the second is the plug-in approach. Copy-paste means, so the biocontainer is a container-based system where all devices are standardized, and this container-based system comes with its own cleaning purification system. So it's a clean room system, which is based on containers. So that means the containers can be shipped anywhere. And the containers are first installed in Germany, in Mainz. The process is implemented for manufacturing. And then the container can be shipped to another place, which is prepared to take up the containers. And once the container are installed there, the process can be copy-paste, by digital means.
Yeah, it's fascinating. I get the concept. I guess what I really want to know is it happening already, and where? In Africa in particular?
Yes, we started to identify partners for this concept, for technology transfer, with the help of the WHO. We identified at the moment 3 potential sites. The first site is in Kigali, in Rwanda. We had already the groundbreaking for our first biocontainer site in July this year, and our goal is to ship the first biocontainers, latest beginning next year, to ensure that in 2023 we can already get the first mRNA doses produced in Africa locally, according to the best possible technology that is also available in Europe or United States.
That's very interesting. So you mentioned Kigali, Rwanda. What other sites in Africa are you actively involved in now?
Yeah, we plan further sites, Pan-Africa. The next sites will be most likely in Senegal, as well in South Africa. But the idea is to establish really a pan-African mRNA manufacturing network, yeah, which is fully digitalized and comes with cutting-edge technology. The beautiful aspect of the BioNTech system is that technology transfer does not only mean the transfer of the technology that we have now, yeah, but technology transfer can be adapted to all future technologies. And what we actually want to accomplish is not only to ensure that future generations of pandemic vaccines could be produced, but also vaccines against infectious diseases, which are extremely relevant in these regions.
No, thank you. I want to get to other diseases in just a moment. We don't have so much time left, so I'm quickly going to get to one other issue that I think is important. What you've just said about addressing these issues of equity in vaccine availability, it's very important, and it does suggest you both are very committed to sort of the global public good.
The task that we see and the responsibility is to make this technology available globally, yeah, because we have to understand that this technology was not only developed in the companies, but also with contribution of many scientists worldwide. And our goal, our vision is to make our technology available as broadly as possible. And I believe there is no reason, yeah, and no challenge, no economic challenge to address that. We want to make this technology available at cost. So that means for us, going to regions like Africa is not about business, it's about social responsibility. And we see social responsibility also not as a kind of giving something, but also interaction and collaboration with the people at the site. And it's part of our vision, it's part of our mission, yeah. And I believe it must be also part of every company developing new technology that could benefit mankind to make this technology auch available in a reasonable way.
I think people will really share that sentiment, Ugur, here at WISH. We don't have much time, so briefly, if you would, Özlem, just tell me what you're most excited about now, looking forward. Let's say that, thank God, we've found vaccines which are capable of combating the worst of COVID, and millions of lives have been saved, and that is terrific. But nobody believes that, pandemics aren't going to be a feature of the future. So what are you working on right now? There's talk of malaria, there's talk of, in the long run, cancer. Do you believe that, vaccines can address these most problematic diseases that we humans face? And how optimistic are you?
We are very optimistic because we believe in the potential of the immune system, and this is what our company is about, redirecting the immune system to address all those diseases with new technologies, and mRNA is only one of those we are pursuing in our company. And we are particularly excited about the fields in which we are currently working clinically, or will be working soon clinically. On the one hand, these are our cancer medicines, including vaccines, where we have 15 different compounds in more than 20 clinical trials in testing, and see that we have clinical activity with our cancer vaccines, therapeutic cancer vaccines, but also other platforms like our engineered T cells. Cancer is one. The other is going broader in infectious diseases, which includes difficult diseases such as malaria, tuberculosis, HIV. In malaria and tuberculosis, we will start clinical trials within the next few months, not only introducing for the first time mRNA as a vaccine technology, but also leveraging our deep domain expertise in immunology. So pursuing new antigens, which speak very deep to the Achilles heels those diseases or those pathogens might have. And
So
there is also
it's not fanciful then to think that in the coming months, maybe years, we could be looking at vaccines for malaria and TB. Is that really what you're saying?
Yes, that's what I'm saying. There is already, as you know, a malaria vaccine approved, which shows the blueprint how to develop malaria vaccines in regulatory and policy terms. And that's an excellent platform to now come with new vaccine targets, with new vaccine technologies. And as I said, we will enter clinical testing within the next couple of months and treat the first subjects.
Well, we definitely want to keep very close tabs on that. We have to end in just a moment. I'm going to end with a personal question. I mean, it's so nice to see the two of you side by side on screens. Obviously, you're not in the same exact location, but you are an amazing couple. You know, you've been married for a long time. You founded BioNTech together. You work side by side every single day. Many of us know, you know, marriage is hard enough without also sharing a business vision, sharing day-to-day running of a company. As the stress, you know, is obviously mounted as you become more and more successful, how do you manage to keep it all together, to stay sane and stay together?
You know, there is a saying that quarrels come based on boredom. And we are never bored. So that's a good platform for a relationship. And we see it as a blessing that we have partners who share the vision and who share the interest. Developing innovations is about partnerships. And this starts basically in ours, but can be extended. We have a great team, partners who support what we are doing.
Ugur, would you recommend heartily working side by side with your wife?
Yeah, absolutely. This is a privilege, and I'm extremely grateful that I found my partner very early in my career. Yeah. So that we can share our professional time, yeah, have someone whom I 100% trust and who complements every aspect that is missing in me as someone with whom we can achieve more things than alone. And I think this is the most important aspect of teamwork, yeah, to achieve things that no one alone can achieve. And this, as Özlem said, goes way beyond the partnership with Özlem. It goes to the partnership with our team members, but it also goes to the partnership with all the partners with whom we are collaborating at the moment to accomplish, for example, the setup of biocontainers in Africa to enable that we can develop mRNA vaccines against tuberculosis and malaria and make them available to people. There are so many partners involved, and together we can accomplish things that appear to be impossible, yeah, if we align on the vision.
Well, you know what? I think that message of stronger together is a great way of ending this interview. It's been such a pleasure to talk to you both. You've achieved so much, and obviously you've given us an indication of what you hope to achieve in the future, which is very exciting. So I thank you both very much for joining all of us here at WISH. Thank you.
Thank you, Stephen.
Thank you for it.
It was a pleasure.
That was Professor Sahin and Professor Turecci speaking to my colleague Stephen Sacker. Now I'm delighted to welcome to the stage the participants for our first panel of the day, Fulfilling the Promise of Precision Medicine, the Essential Role of Biobanks. If I can please ask our panelists to join me here on the stage. free to sit wherever. Yeah. It's my great pleasure to welcome onto the stage Dr. Nala Afifi, the Director of Qatar Biobank, Dr. Karen Saguyasan, Managing Director for International Biobanking and Education, Medical University of Graz, Dr. Zisis Kozlakidis, Head of Laboratory Services and Biobanking, International Agency for Research on Cancer, the World Health Organization, and Dr. Alison Parry-Jones, President-elect for the International Society for Biological and Environmental Repositories. Thank you all for joining us here on the stage. Dr. Kozlakidis, I'd just like to begin with you. If you can help our audiences understand what biobanks are and what precision medicine is.
Thank you very much, and thank you for your invitation and the organization of this conference. It's fantastic so far. Now, precision medicine is a very simple concept, although it's quite difficult to actually enable in action. So we're talking about getting the right medication to the right individuals at the right time in the right dosage, so that they can be able to combat whatever illness they have, or if we're talking about diagnostics, diagnose whatever illness might come and prevent it from appearing. Now, in terms of biobanking, in order to enable this precision vision, we have to test a lot of theories. And in order to be able to test theories, we need biological samples. So this might be blood samples or saliva or urine, any type of physiological sample that will be used in, for example, diagnostic development. Our technology allows us to do this in very high throughput volumes. So we don't test one individual sample at a time, but maybe 396 or 1,000. And you need a standardized way of providing all these samples, and that's where biobanks come in. There are the methodology of providing research-ready or clinic-ready standardized material in high throughput volumes to progress science.
So tell me, how easy is it then to collect this information or data from low and middle income countries especially?
Yes, we're straight into the deep. Eh? So you're absolutely correct. Biobanking is not about only the physical samples, it's also about the related data that comes with it. And the legacy systems that we've had in a lot of low middle income countries depend on disease-specific initiatives that have taken place. So for example, research on HIV in South Africa or malaria in West Africa. And we have good expertise for this particular niche of clinical investigation, but not necessarily a digitalized health record that connects the dots into the patient journey through the healthcare system. And that's really where the challenge is. We get little glimpses of data, and often high quality data, so we don't have to disregard the quality side, but it's not connected in the same way that you find the data connected in Western Europe or North America, and therefore you can make a lot better use of that data and start saying, well, I understand what's happening now, so maybe I can look back in the future and use that information to predict for others.
Why is it so hard to digitalize in these places? Again, is it access of technology?
Yes and no. So up to a point, we're talking about basic infrastructural challenges. Still, we don't have a reliable electricity coverage in many places and a reliable communication coverage. But it will be wrong if we just focus on the infrastructure, because we do have solutions, especially through COVID, that have emerged and make a good use of digital technology. So you can use your phone now to record physiological data that remotely that you would do previously in the clinic. So there are solutions. It's just being able to find context-specific local solutions for the local problems that allows to feed into the methodology that already exists.
I'm just going to bring in Dr. Sargsyan. I mean, this must be incredibly frustrating then, because as the doctor was saying, it is quite simple. You could use your phone. However, in some of these low and middle income countries, it's just proving so incredibly difficult to digitalize.
Yeah, it is really very challenging, but not only about, it is about the infrastructure, it is not only about the standardization, it is about the mentality too. And I don't speak about mentality in low and middle income countries and the Western world, but I speak about the mentality of doctors. Doctors feel themselves, and I'm myself a doctor, I may say that, as Picassos of the medicine, so they don't want to do a standardized job. So the data that would be need to do a precision medicine should be highly standardized. So, you know, that you can put them in zeros and ones. And if we don't use that kind of data, but we use very nice texts that are written like essays, then it is a very big challenge. And we see that in the Western world, doctors are a little bit less tend to write essays as in the low and middle income countries. And I have very good example of biobanking activity that we are in together with Dr. Kostakidis in Armenia. And it's great amount of data, but to standardize that data is very challenging.
Why is it that cultural difference? I mean, given perhaps in low and middle income countries, they would have more patients, say, and therefore, would it not be easier to just standardize?
Especially we speak about cancer. Sure, you can look through the WHO classification and WHO data where the cancer is located, and they would have enormous amount of cancer, especially stage 3 and stage 4, and that would be for sure easier to standardize, but that standardization means a doctrine to come down and to put the borders and to put people in frames, which is not so easy.
What would you say to those doctors then?
I am speaking to them and they don't like me and they say you are. But we have managed to show the effectiveness of that, especially in genetic cancer diagnostics, for instance, that as fast and as standardized you are diagnosing, and easier and as more effective the treatment may be for the patient. So I think we need to speak with success stories to those doctors and motivate instead of push down, then it will function. So we have an example that it's functioned very good with successful stories, but you cannot put there the equality. So if we speak in the research or in work with equality, I mean, think over, if we speak about the equal number of any kind of people in one big cohort, if we look for the breast carcinoma, well, in Graz, we are collecting already 30 years and have 2 men only. If we would wait to compare men with women, we would never be able to do a mama carcinoma research. It's the same with skin cancer. If we would stay and wait to have black-colored people, the same amount like Caucasians, that would be rather difficult. So what I want to say, as in mentality and standardization, as also in research, you cannot put a stamp. It's case-to-case decision, but success stories always help.
Well, I want to ask Dr. Parry Jones about that. Can you help our audiences understand what some of those success stories are? What have you seen in your research?
Well, I think it's important to remember, I work in cancer research in my day job. So I run the Wales Cancer Biobank, and I think it's important to remember that cancer isn't a single disease. You know, we are talking about a whole myriad of diseases, even within breast cancer, as we know. You know, there are multiple different pathologies there, and I think one of the important things about a biobank is just having the quantity of samples, but also the quantity of data. But it does go back to that standardization. If you don't have standardization, then you are not comparing like with like. There was a very worrying statistics a few years ago that a huge number of cancer research was actually not reproducible because they just weren't putting in the publications any reference to methods and materials, you know, when they were using samples. So from that point of view, although not everything is intrinsically reproducible, I understand, but from that point of view, you can't actually compare like with like. And when you're building a biobank, you need that. Or a resource rather, not a bank. That always sounds very sort of insular, but it's a resource to enable the research that goes on. So you do have to be very aware of this, and I think part of the issue that has already been mentioned is around being able to collect this data, source data. So being in the research community, it's sometimes very difficult to collect the data that is in the clinical realm. It's about trust, and that's trust from the patients to be able to allow data to be used in this way for research. But I mean, there are various examples. I mean, you can look back if you have a well-characterized, large enough group of samples. It gives you the chance, for instance, to go back, compare it with maybe molecular data, and then you can start to unpick why a patient or two patients who maybe have tumours that look identical down the microscope actually respond in very different ways to the same drug. So then you're starting to unpack what is the genetics of this, or the genetics of the tumour, and that's where you start to get the newer treatments that are more focused on a genetic outcome rather than focusing on, is it breast cancer, is it colon cancer, what is the mutational status of this tumour.
When it isn't standardized, or people do it in a way that you know, as you say, sort of a few years ago, you saw some of those challenges. How much does that set things back?
Oh, I mean, it can be enormous. I mean, if you're looking to do studies to evaluate clinical outcome, sort of including risk, diagnosis, prognosis, and if you are basing drug discovery, biomarker discovery on samples that you have no idea how they have been curated and collected, etc. We've all heard the garbage in, garbage out, and that's the danger that you have, is if you are basing studies and you're actually using what are basically unknown quantities, then can you actually be assured of the outcome that comes out of that? So, you know, I'd hate to suggest that we are basing biomarkers or anything on erroneous data, but you just don't know. And I think one of the things I think that Giles said earlier is about not worrying about things that you can't control. And not worrying about them is one thing, but you do have to document them, and then you can actually take them into consideration later down the line. Yes, there are all things that we can't control, but you have to know about them to be able to take them into account.
Fascinating. Dr. Nala Afifi, tell us about the experience here in Qatar and its biobanks. How does it work here?
So from the start, I would like to refer at the beginning to the title of the session, to promise to fulfill the precision medicine. So to fulfill the precision medicine, it's need the changing of the mind of all the infrastructure in the medical field, from the classic way of diagnosis and treatment to the precision and targeted solutions to be implemented for the participants or the patients. So in Qatar here, we have a great example that shifting and the infrastructure here starting from the presence of the academic health system strategy since 2008, where we have involved all the partners from the academia, as in Qatar University and Hamad bin Khalifa University, who provide healthcare programs, and also the healthcare providers like Hamad Medical Corporation, Primary Health Corporation, Sidra, and all, even the commercial and the private sectors, in addition to the research institutes. So all of those are working together in the same ecosystem, health ecosystem, targeted to one direction. In addition to that, we have in Qatar here a unified single medical record system, and the clinical record system with Cerner, which is used by the public and the private sectors. And finally, that we have up-to-date state-of-art infrastructure of research centers, either in Hamad bin Khalifa, as I mentioned, or in Qatar University, in addition to the biobank.
So you say that, that it's all part of one ecosystem. Do you think that Qatar's small population helps that as well?
Exactly. That's why it is very important with the number of the population, and all the partners who provide the service are on board. So in Qatar Biobank, when it was established in 2012, 10 years ago, that to provide the service, to be like a center of excellency for providing high quality data and samples for researchers and medical practitioners to for discovery and implementation of the precision medicine. So since the start of the biobank, the vision of the decision maker for implementation of precision medicine is here. So in Qatar Biobank, we found that after 10 years and after recruiting 36,000 participants in our cohort longitudinal study and follow them after 5 years, we found that we have a great role in implementation of precision medicine. As everyone knows, precision medicine starts from preventive stage, like before the participant or the patient develop a disease. So from our data, as we collect clinical and comprehensive clinical and medical data, we found that like 20% of our population are pre-diabetics and 25% are pre-hypertensive, meaning that within few years, they would develop diabetes and develop hypertension. So this is the correct time to intervene and to prevent. It's only changing the lifestyle, changing the diet, more exercise will prevent them to develop the disease. So it is, and they decrease the burden on the health system in Qatar.
I just wanted to come to you, Doctor, and just pick up on what Dr. Afifi is saying there, because success story, Qatar is a smaller population, it has a more organized ecosystem. What do larger countries, what does, a country with a population of several hundred million, for example, do?
They tend to work in islands of excellence. And then the challenge is, how do you connect these dots to create the ecosystem that we see in Qatar? We have loads of good examples for these islands of excellence in many countries in the world. So India with the Tata Medical Center, in Brazil with Barreto's Hospital. The list is long, we can go through. The question is, how do you connect the data to get the added value at the population level? And that's where our challenge is at the moment.
How do you? We'll try and resolve it here on the stage.
Well, COVID has been a great example that actually we can do it, maybe not entirely, but if we're focused enough for data parameters that give us value, then we can connect those elements and bring them together under a single roof for research purposes or clinical purposes. We do have to deal with a lot of legacy systems. So it's all nice to say there is this new technology, let's introduce it, but it has to work together with all the other technologies that have been introduced over the past 30, 40 years. And in order to be effective, it has to work in a seamless way. And that's a lot more of a challenge than simply introducing new technology.
I'm going to open the floor up to the audience. We have about 10 minutes, so I can take a few questions. There are some mics going around, and I see a hand going up here. If we can get a microphone just to the front of the room, that would be great. And before the microphone gets there, Dr. Parry-Jones, I just wanted to ask you, and I just wanted to pick up on this comment about COVID is a good example, but has COVID also been disruptive for your research funding and that kind of thing?
I mean, it has been mainly because the patients have not been coming into clinic, so we haven't been able to consent patients to collect samples. So it has made a change in that way. It has actually forced us to look at more innovative ways of contacting patients, of being able to collect samples. I think it's also helped, it's raised awareness within the general public of what a biobank is. It may not have been called a biobank, but it's certainly raised the understanding of why collection of samples is necessary to be able to look at a range of diseases. So in some ways it has helped us, and in other ways it has clearly hindered us.
Hindered, yeah. I'm just going to go to the question at the front, if you don't mind standing. Perfect.
Doesn't work. Oh,
there you go.
Thank you very much for a very, very interesting, illuminating discussion. What I want to do is just give a laboratory perspective, precision medicine. My name is Walid El-Wali, so I'm I work in the laboratories of the Hamad Medical Corporation. I'm also the acting head of the Division of Diagnostic Genomics. Diagnostic Genomics Laboratory sits at the epicenter of precision medicine. I just want to share very quickly our experience in Qatar. Basically, our laboratory consists of 7 wonderful clinical scientists and 60 wonderful technologists. And what we do, we provide basically, in terms of our role, we feel we're very instrumental in terms of the diagnosis, prevention, in terms of prenatal screening, and also premarital screening. And essentially what we do is basically, apart from real-time PCR, gene sequencing, next generation sequencing, and we're going to embark on whole exome sequencing. In addition, we do microarray FISH and also karyotyping. We feel that basically our function is in the diagnosis of first mutations at germ level to identify things like inherited diseases, inherited cancer, and also at somatic level for solid tumors and hemato-oncology. I want to say, since we're talking about biobank today, that we have a very close, fantastic relationship with Qatar Biobank, and there is a big project, which is the Q-chip. More than 6,000 of the population have enlisted in that project, and this will immensely improve our diagnostic capability. Thank you very much.
Can I ask you a question?
Sure.
What advice then can you give to countries that are struggling with their data collection?
Well, very important is that they should have bioinformaticians, very crucial. They should have a very strong infrastructure and robust information governance to ensure that it is strong, reliable, and robust. I think it's very important to start in the right way. It's really following solid, robust governance. That's what it is. And if they don't have that, I'm sure there will be other countries that will support. And Qatar is a fantastic example.
I mean, Doctor, just to pick up on that point, though, sometimes, as you say, the governance is weak, or you have very old traditional systems that are hard to break.
Yes, and the opportunity then lies in the partnership. So we can have twinning programs, for example, like we do with Armenia. You can have mentorship institutional programs where institutions or regions can adopt others and help them on the way. And I think the president of Tanzania yesterday mentioned similar activities in this manner. I think one of the complexities that we face is how do we, all this information that we collect, we integrate into the system, how do we then transmit it back to the patient? Because that's where the whole journey starts from. And this is something we're dealing in terms of the complexity, but also something that is very actively a positive message. And again, coming back to COVID, it has been a great example on how people are willing to participate in this active ongoing research and they see the benefit coming back to them or to their children or the immediate family.
Dr.
I can take the,
yes, please, go ahead.
the talk from Dr. Zisis about how we transfer this to the clinical and to the patient themselves. Like one example which we are so proud of it is that the findings of the whole genome sequence of the first 6,000 participants, like one project which was published under the publication strategy led by Qatar Genome, that a project with where they look for the genes like BRCA gene from Weill Cornell with Dr. So that's why when they identify that there are participants, 20 participants, they carry the gene, and they are not, from our data, they are not diagnosed as they have cancer, breast cancer. So what to do with these participants? So a new project to complete the loop from a research project to the implementation of in the healthcare system. So, from Qatar National Cancer Institute, like we identify like that she has to work on identifying these persons, how we will identify them, and to provide them with the genetic counseling and to double check and confirm the results of this participant. It has to be confirmed at the medical institute. So we sit together for more than 1 year to study the process to complete the loop between us as a biobank, where the participant to contact them again, and to sign a consent and convince them to give blood samples, which will be analyzed in the hospital in the CAP accredited lab to double check and confirm the results that they are carrying the gene. And then after that, call them and referring to the specialized healthcare clinic with the genetic counsel and contact their families. We were so successful that we complete the loop of these 20 participants. 12 of them, they came to the biobank, they signed the consent, and they give the blood. It confirms the same results which is found within the research concept, and that they follow them and their family in the hospital. So this is one example of how we implement the precision medicine successful, and to get the fund from Qatar Genome, to get the commitment from the providers, the physicians, and also from the researchers. All of them, they are working together within the biobank team. So I have to acknowledge this project and the success of this project in Qatar.
That's wonderful. Dr. Sargsyan, did you want to comment on that?
Actually, I wanted to bring another very successful example. It's my own. I mean, I am genetically affected by familiar Mediterranean fever, but I'm not ill. And I am going to check every single time I'm getting high fever. And I know that I have that gene and it can become an illness. So it is awareness of a patient. In my case, I'm a patient, not a doctor. So, and it helps so much to care for your own health with understanding of your genetic background. So I wanted to prove the point that Dr. Nalla brought with my own example.
But do you think it helps you're a doctor, so therefore you can act as a patient?
I cannot answer that question because I'm myself a doctor, and the doctors are the worst patients ever. So I think they don't like me at all, but it helps anyway to know, because they are aware and they will not miss the early diagnostic stage when you can do something. Especially, I mean, not for FMF, but for cancer, it's very important when it is diagnosed for the outcome. And we see in the statistics worldwide that as early it is diagnosed, as better the outcome is. So it's very important to know the information, especially as the question came with the huge amount of data. Well, first you need to digest that data for the doctors, because doctors are not used to raw data, being honest. But if it is digested in a form of the biomarkers that doctors know as a risk factors and so on, then I think it helps so much for each single patient, and then, because of that, the whole health system.
Yeah, and Dr. Parry-Jones, you can probably attest to that. I mean, you constantly hear early diagnosis, go and get checked up, whether it's breast or cervical or prostate, any kind of cancer. Often people are encouraged to go and check if you sense something is wrong.
Oh, absolutely. I mean, early diagnosis is key. There are far better outcomes, I would probably say, for most cancer patients, if not all, if the earlier it's detected. So, yes, I mean, I would certainly echo that, and the screening programs are a very important part of it. And again, as a biobank, if you can actually collect samples from those screening programs, the most valuable set of samples, valuable not being monetary value, but scientific value, is if you can collect right from the beginning of before a diagnosis, right through the patient's journey, and even post-mortem samples, if you're able to collect those, then you can really see what is happening, talking here maybe specifically about solid tumours, but what is happening throughout that whole process of the cancer.
On that note, go and see a doctor if you sense something is wrong. And I'd like to wrap up this panel. Thank you so much to my panelists here this morning. And that brings to an end this second day's opening panel. Of course, I encourage you all to go to the Zulal Wellness Center, which is just on this floor, I believe. You can go there and get a massage or get some reflexology. They're waiting for people to come and check them out, so please go there. And don't forget, at 11:15, our next panel on the health and well-being of migrant workers with my colleague, Stephen Sacker. Thank you so much for joining us here again today. Thank you so much. Thank you.